Hope we will have some time for questions and comments. So our next speaker, Dr. Amy LeBlanc, is a researcher at the National Cancer Institute, and she's going to talk with us about naturally occurring tumors in our pet population as a model for improving cancer care.
Thank you. Thank you very much for the invitation to be here today and the support from the Academy and the forum organizers. Very much appreciate the chance to come and tell you what the National Cancer Institute has done in investing in the study of spontaneous canine cancers as models for precision cancer medicine, and how we can learn from our companion animals and how that can improve the lives of their lives, but also ours as well. So what I want to do just in our time today is give you an idea, if you're not already familiar, with what the concept of comparative oncology is and how that approach can both improve and inform the cancer research and drug development process, and put the emphasis on precision medicine in mind as we go through this today. Several of the speakers today, and I'm sure I wasn't here yesterday, but yesterday I think has really teed this up nicely for me, so I appreciate that. And I'll also give you an idea of the program that I run at the NCI, its infrastructure, its goals, give you some examples of the trials, the clinical trials that we run in dogs that live in homes as pets. They are patients, so they themselves are patients, just like humans are thought of as patients. And also give you an idea of some new NCI-funded initiatives in comparative oncology that may not be widely known about. So there's been a lot of interest and enthusiasm around investing in this concept in a significant financial way on the part of the NCI, so I'll definitely tell you about that as well. So the Comparative Oncology Program is an intramural NCI, so we are within the Center for Cancer Research, and the branch that we belong to is actually the Molecular Imaging Program. That's because my scientific background is in comparative cancer imaging, and so that's where we sit, along with other intramural and extramural areas of the NCI. It's a lot of alphabet soup, but we work a lot with both our intramural scientific partners and collaborators here in Bethesda and up in Shady Grove and Frederick, but also we work a lot with some of the extramural entities as well, specifically the DCTD, which is the Division of Cancer Therapy and Diagnosis. So I'll tell you about one of the trials that we did with DCTD funding and support that has really then given rise to several of the new funding initiatives and new opportunities for clinical trials that I'll discuss later on in the talk. So the Comparative Oncology Program goals in general are not in any particular order, but initially it's important for us to really advocate and support the integration of pet dogs that naturally develop cancer within the context of their natural lifespan and environmental exposures into clinical trials through a clinical trial infrastructure that we centrally manage and maintain, which is called the COTC, the Comparative Oncology Trials Consortium. That affords access of the drug development community to these dogs. So these are pet dogs that develop cancer, and their owners seek care through veterinary schools that operate veterinary teaching hospitals that have oncologists like me there to take care of the clinical caseload, but also we have a teaching and research mission as well. And so our clinical trial infrastructure marries those groups of people. So folks from the drug development side that are interested in understanding how they can study these patients to advance their drug development pathways. We also have a laboratory, so we are an intramural program, and we have an intramural laboratory that is currently focused on understanding metabolic vulnerabilities in metastatic progression. We use dogs that develop osteosarcoma, which is bone cancer, that has a very high rate of metastasis, and it provides a really nice opportunity to understand the complexities and the biology of metastatic progression. And it represents an opportunity to study a very common canine disease and advance that knowledge for an orphan pediatric malignancy. We also, because of the background that I bring to the program, have an emphasis on developing molecular imaging tools that help interrogate the findings in the laboratory and bridge some of that discovery into the clinical trial setting. And then we have a significant amount of resources as well: cell lines, reagents, assays, tissue microarrays, biospecimen repositories, et cetera, that we then provide stewardship for and offer that to the greater cancer research community. So for those of you who aren't familiar, there are a lot of remarkable similarities in the cancers that dogs get and that humans get. This is from a really nice paper that my friends Josh Schiffman and Matthew Breen wrote recently, really sort of giving you the big picture overview of where things can be shared between dogs and humans. There are millions and millions of dogs that are getting a cancer diagnosis each year, and the incidence is likely much higher than it is in people. Recognize that some of these statistics are not as strong as they could be, and so that's another sort of big initiative in the comparative oncology community to really capture as many diagnoses as we can and help characterize these types of cancers as much as possible to then understand their translational relevance to human patients. There are some diseases that dogs don't get that are very common in people, or at least they get them at a much lower frequency, and some of that may have to do a lot with reproductive status and environmental exposures and things like alcohol consumption and cigarette smoking as factors in cancer development. So, dogs are not going to get a lot of lung cancer because they don't smoke. And if they're reproductively altered at an early age, that will reduce some of their incidence of certain reproductive cancers. So it's not always a perfect match, but there are many, many types of cancers that are very nice models for their human counterparts. So this is the clinical trial infrastructure that my program oversees. We function because we have such amazing collaborators and site investigators at these 22 veterinary schools in the US, and we have one Canadian site at Guelph. And so it's because these people allow us access to patient populations through their veterinary teaching hospitals that we have the ability to conduct multi-center, very high quality clinical trials in dogs that naturally develop these diseases. And this is a fairly famous figure for those of you who have read or know anything about comparative oncology. The comparative oncology program was actually founded by Chan Khanna back in 2004. He's also a veterinary oncologist and had trained at NCI in a postdoctoral fellowship and then was given the opportunity from the Intramural NCI to invest and grow this program. And this figure was from a paper that he wrote that really just sort of crystallized where we believe that the naturally occurring cancers in dogs can really inform at multiple levels during sort of the classic drug development paradigm, but also important to recognize that there's bidirectional benefit. So what we learn about studying cancers in dogs, from my perspective, is really the main intent is to inform human health, but also we learn about these diseases for the sake of the dog as well. And we offer that knowledge and experience back to the larger cancer community that more and more includes people who care about understanding the diagnosis, management, and prevention of cancers in dogs for the sake of them as family members. So this is a slide that Matthew Breen from NC State gave me, and so he told me that every time I have to show it, I have to give him credit for it. So this is Matthew Breen's slide. And I think it just, again, it hammers home one of the big concepts around comparative oncology clinical trials, in that you, within the operation of these trials, have the opportunity to collect a significant amount of biologic data from these patients. So we, of course, operate under the guise of animal care and use standards, and every single one of these trials is subjected to significant review and rigor from the Animal Care and Use Committee's standpoint, not just for animal welfare concerns, but also for scientific merits and for asking very critical questions about why you need X number of dogs to conduct this trial and why you need X number of patient biopsies or tissue samples or blood collections. All of that is really rigorously reviewed for the ethical mandate that we, of course, are bound by, and we're happy to be bound by that. But one of the comments that I think I'll make here is that specifically when we interface with our human clinical trial counterparts through either CTEP or NEXT or DCTD or the clinical trial networks that are operated by the NCI, they consistently remark about how we in the comparative oncology world can collect amazingly high quality samples and repeatedly, and we almost never have missing data points because there's a refusal to collect this particular sample. And so we, I think, really perform quite at a quite high level, and we're really proud of that because it means that we generate very biologically rich data that really can inform on multiple different levels. I'm just going to give you a few trial examples and then get into some new initiatives that the NCI is investing in currently and will continue to fund. So this paper is actually from a funded study that was supported by EMD Serono, looking at a novel immunocytokine, NHS-IL-12. This was a clinical trial that was conducted back in 2007, 8, 9, around that time. And what's interesting about this trial is that we gave this immunocytokine, this is a fully humanized immunocytokine, we gave this to dogs in a single dose, and then eventually we did some multi-dose studies. And it initially was designed as an all-comers trial, so any dog with any solid tumor could be eligible as long as they had good performance and met other criteria. And the company at this point was sort of poised to dump this drug. Because they weren't really sure what to do with it, whether or not it was, this is sort of before a lot of immunotherapy kind of caught fire. And the data that we generated in this trial gave this particular company an efficacy signal that they were not anticipating, and also gave them and the NCI enough enthusiasm to generate an IND. And now this is a drug that's in phase 1 clinical trials at the NCI and the clinical center. It also informed a lot on responding biomarker selection and how to actually write a good IND for this agent in humans. And so repeatedly this comes up that this was one of the really, probably the best examples that early on gave value to the comparative oncology approach. This is a DCTD-funded trial where the dog with lymphoma was enrolled and was simultaneously randomized across 3 TOP1 inhibitors with the express intent of trying to figure out which one should be prioritized over the other 2. So this work actually is one that will fulfill a true cross-species analysis. So these agents were studied in mice, they were studied in dogs, and they're going to be studied in people. And as this work continues, we'll be able to retrospectively say, you know, where did the dog's value really lie? And I can already tell you that the dog data predicted that one of these agents was distinctly different and more biologically active than the other 2, and that was not data that came out of any preclinical mouse data. So that was another great example of where we really informed the path. And then, just in the interest of time, I'm going to skip through this. We do have an oncolytic virotherapy trial that's open, so we do, play in not just the small molecule space, we get into biologics and other immunomodulatory therapies. I've already mentioned a little bit about osteosarcoma, so I'll kind of skip through that. I think a couple of main points here that when we do these types of trials, when we work with a sponsor and put a trial together through our clinical trial network, the first thing that we need to make sure is that that trial is being done to meet a specific need that's unmet in human drug development. So we really spend a lot of time making sure that the data that we're going to generate will really impact or fill a gap that's currently unmet. And a lot of times we're not really looking at efficacy as a main signal, although that is always a really nice thing to see. We're more biologically rich in that we're trying to define target modulation and PK/PD relationships, and that the FDA is very much aware of these kinds of data and has seen comparative oncology data in several IND filings over the last 5 to 7 years, and they view it in context and view it as important but supplementary, and have said on multiple occasions that it will never derail the clinical development. So they, adverse events can be characterized as expected and never really fear that it would result in a clinical hold if it was linked to an open IND of a human drug trial. So I think kind of moving forward, what do we think that this concept can do for the whole idea of precision medicine? In my opinion, I think that the biggest thing that we have to address is what the genetic landscape is of canine cancers, because comparative oncology has not really received any significant amount of funding until really recently to help understand the types of cancers we see in dogs past the clinical observations that, yes, they develop naturally in the context of intact immunity and environmental exposures and a natural species lifespan. That descriptive information is valuable, but it doesn't really get you to where we really need to be, which is to say, what percentage of canine soft tissue sarcomas harbor a RAS mutation. We don't know that. We will know that soon, but that's, I think, the key question for us to be able to participate in this conversation. We also need to have access to therapies that match the genetic abnormalities that we will eventually uncover and describe in the dog. And we need better analytical tools and expertise. That sort of goes to the educational piece that's been brought up this afternoon. I think access to biologic samples and clinical and demographic data is relatively straightforward for us because we're not bound by HIPAA and other sort of privacy sort of situations that become real issues on the human side. So I think there's a lot of opportunity there. Recently, just to get you up to speed on what the NCI has done from a funding standpoint, a first and major step was that last year, the NCI gave out 8 awards totaling about $4 million through Cancer Center Supplements to characterize the molecular landscape of selected canine cancers. The goals of that administrative supplement were really to, really at its core, generate sequencing data across selected canine cancers to understand mutational load and really understand the immune components of those tumors, which really then led nicely into the next step, which was to create an RFA to investigate utility of these canine patients for immunotherapy development. So these awards just got announced just in the last few weeks. The long-term goal of providing this funding for the NCI was to really help establish the suitability of dogs with cancer as models to study both single and combination immunomodulatory agent strategies, and then really to expressly translate those findings into human trials. This is how it's going to work. So these were 5 awards totaling about $15 million. This was, again, it represents really a significant amount of funding from the standpoint of comparative oncology. My program will be assisting the U24 Coordinating Center awardee, which is at the University of Pennsylvania, with coordinating. So there's actually only 5 UM1 awards. This diagram has 6, but only 5 were chosen. And then these awardees are studying glioma, metastatic osteosarcoma, melanoma, and lymphoma within the context of canine trials carried out at their institutions. Another thing as far as financial investment, so the NCI DCTD has invested multiple millions of dollars into my program to help sponsor a series of phase 0 studies in canine cancers. So we will be working with our COTC member institutions to basically generate a series of short, straightforward, sort of clean biopsy-treat, biopsy-type trials where we will be studying dogs with peripherally accessible disease to help answer very simple and prioritization directed questions about selecting a lead agent, understanding PK/PD, tolerability and efficacy, and sort of short-term exposures over 2 to 3 weeks, and really drilling down on target modulation and things like methylation profiling and synthetic lethality. We also started a brain tumor initiative for those of you who are interested in knowing more about that. There's more to say about this is a research consortium, not just a clinical trial consortium, so if any of you are interested in knowing more about this, just let me know. And this just really sort of captures what the Brain Tumor Consortium's structure looks like, and that we organized a whole group of people who, across many, many disciplines and walks of life, were organized into working groups, and now individual projects are in progress across all 5 of the working groups listed here. And so I think today really represents a really nice extension from a previous National Academy workshop on comparative oncology back in 2015. And so that really helped us, I think, as a community recognize the value of these types of studies, and it has really garnered much of the, I think, the enthusiasm and the funding opportunities on the NCI perspective and others in the pharma and other drug discovery stakeholders into wanting to know more about where comparative oncology can add value. And so I think in summary, for me, I think the success in precision medicine is really predicated on knowing a lot more than we currently know about the diseases we see in dogs. And so we have a lot more work to do on that side, but I think that what I've seen in the last handful of years is that there's been a significant increase in awareness, advocacy, and funding, and really good research is in progress and has been developed in the last few years. And we have a lot to offer the drug development community, so I look forward to what the next several years really holds for us. I'll just acknowledge everyone in my lab and at the NCI and beyond, and thank you for your attention this afternoon.